season to get those answers. And your trial and your questions may evolve as you go along. After the first year, you may kind of revise what your questions actually are or where you're going with the trial. And like, what are the reasons to do a variety trial? I mean, it's obviously something that is going to add to your workload, and it's adding sort of a different type of work where you're going to have to remember to collect data regularly, and it's just one more thing to do at a busy time. And there are a number of different reasons to do it, any or all of which might apply to any given trial. The main reason might be to test or describe unknown varieties, or varieties that are unknown to you. You might want to evaluate a number of different varieties before committing to growing a large crop of a given variety. You might want to find out if there's something better out there than what you currently grow. You might be looking for something that fills a specific niche, like to extend your season a little bit, or like to have kind of a new product with an interesting taste to sell at the market. And I think something that's increasingly relevant in the context of changing and unpredictable climates is if you want to adapt to changing conditions. So you might have a variety that worked very well for you in the past, but now maybe the springs are warmer, you've got some new insects attacking your crops earlier, and you might need to look into some new varieties, or maybe even a different species that would fit the same need on your farm that would be less susceptible to whatever your new problem happens to be. And so where do you get ideas for trials? You don't want to do a trial just because. You want to do a trial because you've got a real question you want to answer. So I've come up with an example just from my own backyard gardening experience, which is that last fall I planted out kind of a very informal trial of a number of spinach varieties just to see what they looked like. And I sort of accidentally overwintered them. And this spring I noticed that there was one variety in particular, Bloomfield Dark Green, that seems to have overwintered very well. And this was in a Quebec winter, so we had frequently minus 30 degrees temperatures, quite a bit of snow, however, and so on. And so I thought, hey, that's kind of interesting. It's nice to have spinach so early in the spring. And I wonder if this is actually some germplasm that does really well at overwintering. But I can't really know based on this particular trial, because I haven't really controlled for any variables, and including this one variable that's pretty important, which is my dog, who tends to run madly through the garden beds. So it's possibly that Bloomfield Dark Green just got lucky and didn't get trampled as much as the others. And I don't really have any replication, so it may just have gotten, been in a better spot in the garden or something. So I can't really draw any conclusions, but I'm kind of interested now. I'm kind of interested next year and maybe trying out a bunch of varieties, controlling it a little bit more, and seeing what I can get, and seeing if I could isolate some germplasm that would be really good for overwintering. And that's, a very simple example based on my very simple backyard garden, but those of you who are producers presumably have scenarios like this all the time with the multiple varieties and species that you manage. So once you've got kind of an idea for a trial or a question, then the next question is where are you going to get germplasm for this trial? What varieties will you try? Where will they come from? What are your reasons for trying these varieties? And broadly in the field of plant genetic resource conservation, we talk about two big repositories of plant genetic resources. One is ex situ conservation, where seeds are stored, in formal institutions and seed banks. And then in situ conservation, where farmers and local people are actually actively managing seeds, saving them, sharing them, and conserving them over time, and that these seeds are also evolving and changing over time. And both of these types of seed repositories are valid sources of seed for variety trials. And you've got the level that you can look to individuals and communities near you, where you've got all these people saving seeds. Here I've got the example of the Atwater Seed Library, which just launched yesterday in Montreal. And they've got a special, they've really dedicated a lot of work to trying to save and disseminate a lot of traditional Quebec varieties. And so that, different local projects like that, or just individual people can be really good sources of germplasm. And then at a more formal level, but also accessible, you have gene banks and institutions that are really dedicated to conserving, like this is their mandate, is to collect and preserve crop germplasm for use in public breeding, for use by farmers. This is their job to do it. And so you have national institutions like PGRC, or Plant Gene Resources of Canada, or the United States Department of Agriculture, who have very massive collections. You have different international institutions, like in Colombia you have CIAT that works on potatoes and a number of other things. You have IRRI that works on rice, like these large research centers. You have, I discovered last year, there's a nonprofit World Vegetable Center in Taiwan that is dedicated to open pollinated and tropical vegetables. And how do you actually access all of these seeds? Like, they're out there, these people have them, but how you get them? There are a lot of databases. Like a good place to start is the GRIN database, which stands for Genetic Resource Information Network. There's one for the USDA Gene Bank, there's one for the Canadian Gene Bank, and there you can see like all the holdings they have for different crops. So if you're interested in, barley or something, you can go see like all the different barley varieties they have. There's a network called Eurisco that puts together all the accessions from different European gene banks. The FAO has a database called Views that has all existing gene banks in it. And then there's one called Genesis, which kind of filters out maybe data that's a bit old and just has like the highest, the most reliable information on current gene bank holdings. And once you've identified what you want, usually you'll write to the curator of that gene bank and put in a request saying, I'd like to try these varieties. And usually they'll ask you why. Like they're not seed, they're not seed companies. Like they want to provide seed for more of a research purpose. And so if you're saying, I want to try this, this pea variety because I'm looking for something with better disease resistance, then they should normally be able to send you a small amount of those for you to try out. And another source is, of course, seed companies. I mean, there's quite a few seed companies that do produce and sell OP varieties. And these have the advantage of being varieties that are actively used, that you can get larger quantities right away. Like from a seed bank or gene bank, you'll get a very small number of seeds that you'll need to maybe increase before you can do like a bigger trial. And normally the quality control in a seed company is going to be a lot more stringent than at the gene bank, where the gene bank, their mandate is basically just to conserve as best they can, whereas the seed company has an interest in really making sure that the varieties are, true to the variety description, that there's a good germination rate, and so on. So like looking through all the different seed companies is a good starting place too. I did some trials with some growers in 2014, and just for, for interest's sake, like I was looking at spinach and Chinese cabbage, and I got a large number of varieties from 4 different public gene banks and from 15 different commercial seed companies. And so you'll probably end up spending some time like combing through catalogs, combing through databases, and kind of narrowing down what it is you want to look at. And once you've got, your basic question and, an idea of which germplasm you want to test, I'd encourage you to think of your variety trial like as kind of an integrated thing. And kind of the core question is like, how much work do I have to do to know what I want to know or to learn what I want to know? And the first thing that you need is you want to know what you want to know, first of all. And so like it might be, which of these varieties is slowest to bolt? Do I like this variety more or less than what I already grow? What do these mysterious varieties I got from the seed bank look like? There are any number of questions. And depending on what your question is, you're going to have a different level of detail and rigor in your trial. Because again, you don't want to do more work than you have to. You want to do enough work to get the answers. And so if you want to look at, what do the samples from the gene bank look like, you might be looking at just putting down more of a general description of how they develop, what they look like, taking a lot of photos, and having a check variety in there so you can compare them to something that you're familiar with. which of them is slowest to bolt. So then you'd obviously want, the date you planted it, the date it bolted. If you could get temperature data, that would probably be good too. And again, a check variety so that you can compare it to whatever your standard is now. If it's a more general question, like do I like this variety more or less than what I already grow, it would kind of depend on why you like the variety you have in the first place. So you might want to look at yield, presumably appearance, the taste, and again to have a check variety in there. Here's an example of two different components of the Chinese cabbage trials that I worked on last summer. This is a trial that Dan Brisebois managed at Ferme-Tournesol in Quebec. And here we had kind of two different subcomponents of the trial. So we wanted to look at heading type Chinese cabbages in fall production. And we had two different trials. The first one was using gene bank varieties. And so varieties we knew very little about other than, what country they came from and when they were collected. So variety might be just, variety 8001, China 1952. Like that would be all you know about it. And so for those, I think we had about 30 varieties. And we didn't bother doing planting a huge amount of any of them. We just wanted to see if any of them was good or not. So there we try to replicate anything. We planted 6 plants of each variety, so a very small number. And we did a really brief kind of data sheet. It was basically does this look good? Does it not look good? And then we looked at also 5 different commercial open pollinated varieties that we had some hopes would be, kind of a more advanced germplasm, subjected to more quality control. And so for those, we repeated each variety 3 times. We grew 15 plants of each, and we collected much more detailed data. So with a given crop, the approach you take will be very different. Then the next thing you'll want to know once you've thought of kind of what your level of detail and rigor is how you're going to lay out your trial. So are you going to repeat different varieties throughout the same variety many times in the trial? How many plants will you have? How will you plant them? Will you be direct seeding? Will you be transplanting? Is there anything you want to look at besides variety? Do you want to try different levels of fertilization, different planting dates, different sites? There's any number of variations on how you set this up. And, there are some basic rules of thumb, I would say, for layout, which is, make sure you treat all the varieties the same way as much as possible, have uniform conditions, plant them all on the same day, take all the measurements on the same day. I'd say the most key one is to always have a check variety, always something that you're familiar with. So in the spinach trial, everyone was growing Bloomsdale as a reference OP variety and Tai as a reference hybrid spinach that most people were familiar with. You want to randomize where you put each variety in the bed. It is really good to repeat each one at a few different places along your bed of your trial. But the more you replicate, maybe the fewer varieties you can do. So you're going to want to find a trade-off. I like to do 3 replicates generally, but sometimes if you want to fit in a few more varieties, maybe you go down to 2. And in the case of gene bank stuff, you probably don't replicate. You probably just do 1 of each. And you'll kind of want to be clear. Here it didn't really show up very well, but I've got kind of an example of an illustration of a layout. So you want to kind of write down how you're going to lay things out and then, make sure that it's very clear to you before you go out to plant them. I've talked about variables, and, variables are anything that varies and that can affect the outcome of your trial. You want to control them as much as possible, knowing that in an agricultural setting, it's very hard to have absolute control. So things like soil fertility, even small differences, if, a big clump of manure fell on one part of the bed, that might make one variety look amazing just because you happen to plant it there. Water availability, light, pests, slowly, so might have kind of some differential damage happening. So you'll control those as much as possible, and that's why replicating can be important, because it can help you kind of see what the effect of variation along the bed is compared to the effect of the variety itself. And yeah, I think the rest of the stuff I've already said, like, you know, make sure you treat everything uniformly, replicate if you can, put things randomly, and gather data in the entire plot at the same time. And so you'll want to be gathering data. You'll need data sheets. And there are a number of different ways, you know, again, ways to set up a data sheet and kind of levels of complexity of data. Like it could be anything from just saying, you know, is this like resistant or susceptible to this insect, or like a yes, no answer. You can rank things. You can say like, is this, you know, on a scale of 1 to 9, is this good? Is this 9 being the best, 1 being the worst? Like where does this one fall? You can have different, there are different scales that exist for resistance to different diseases and pests. You can actually take measurements, like measure things with a measuring tape, weigh them. You know, each of these involves a different level of work intensity. So generally, like with data sheets, you want to keep them as short and simple as possible, but you want to make sure they'll give you the info you need. So again, you're thinking about like what you want to learn from this trial. On the OSA website, they do have kind of like a standard data sheet for trials that you can look at, you can maybe adapt, and they have some tips for trials, so that's a pretty good resource. The link is on the slide here. And here I've just put a sample of a data sheet I put together for spinach. So you have like, you know, the replicate, the variety, and then some of the data. So, you know, the date planted, date seedlings emerged, date of maturity, date of bolting, and then here it was for spinach. And so we're trying to calculate like how many cuttings people got and, you know, how much time it took the plant to regenerate between cuttings. And then the data sheet goes on for a while, but, you know, this is just one example of a layout. And then you're going to be collecting your data. Part of that's going to be written, part of it that's going to be photos. And the rules of thumb for collecting it is make sure you do it at regular intervals. Like you don't want to forget your trial for a month or two because you're going to miss most of the information that it has to give you. You want to be consistent, so you want to make sure you're evaluating things the same way over time. So if you can kind of set yourself some references for what is, you know, what you consider to be like a badly insect damaged plant versus a resistant plant. And you want, like, if it's several people doing the evaluation, you want to make sure that each of those people has the same idea of how the evaluation is happening. So normally how you would do that is, you know, you would each evaluate independently the same part of the plot and then compare your answers and kind of, you know, work together until you've kind of agreed on some norms for how to evaluate things. But, you know, ideally it would be the same person evaluating the whole plot all the time. You know, leave a column in your data sheet for notes because you're going to notice random things that are interesting but that don't really fit into any of your data columns. So just give yourself some room to write down other things you see that you think are interesting. Prioritize, like if you're finding it's taking you a whole day to evaluate your plot and you're trying to evaluate it once a week, obviously that's not going to work. You're going to want to look through and either find ways to do it faster or maybe just take out a few data columns. And I would say one of the biggest ones is to take photos because those can really inform you and help you remember the trial in ways that your notes just won't. And so these are a few photos that Gilbert took at Wild Rose Farm in Nova Scotia from his spinach trials. And I just find they're really great photos. Like you've got this one on the right of the Malsur, the zero flay spinach. And I think that very accurately shows kind of the main characteristic of the spinach. Like you've got this giant leaf. And you know, that's much more evocative and triggers the memory a lot more than just kind of a written description or a measurement. And what you've done on the left is just laid out all the different variety leaves next to each other just so you get an idea of the actual variation in what the leaves look like in the trial. So like, you know, take photos every time you're out collecting data. And the advantage of the photos too is if they're digital, you've got a timestamp on them too. So if you forgot to note some things down, you can look back at your photos and kind of get your dates from those as well. If you're doing trials from gene bank seed, this is just kind of a warning, is that, it's very easy to get caught in the kind of the romantic idea of like there's all these beautiful, well-adapted old varieties in the gene bank. I mean, I would say you can expect, from my experience, like expect, 80% of them to look like absolutely terrible. And that's fine. It's because those varieties, like they're not necessarily meant to be adapted at all to your conditions. They might be for somewhere more tropical. They might, who knows? Who knows what the people who were growing them were selecting them for. Plus, if they've been in the gene bank for 40 years, like no one's been working on maintaining their quality. They've just been working on regenerating them. And so I'd say if you're doing trials from gene bank seed, you want to get a lot of varieties and grow a small amount of each. Because if you just get, 2 or 3, you'll probably be disappointed. But if you grow, 15 or 20 or 30, you'll probably find a few that are fun and that are interesting and that don't really look like anything you currently have. And this is just kind of funny, like this particular, here you see on the right, like my gene bank varieties, mostly looking bad. And like these people that I brought at a field day, and I'm showing them this trial, and then I realized like they, I seem kind of crazy to be proudly showing them this trial, but I think it's actually very exciting to bring stuff out of the gene banks and see what works. And once you've collected all this data, like you don't want to do that unless you're going to analyze it. You want to think from the beginning about like how much work you want to put into this and how you're going to analyze it and what kind of analysis will make the data meaningful to you. The first thing you'll want to do is probably clean it up a bit. Like you'll want to put it into some electronic format, and you'll have all these little notes you've put here and there. A few question marks or whatever. You're going to just need to go through, like, kind of, make sure that it's concise enough that you can analyze it. And you only really need to analyze it enough to convince yourself or the people you work with of the results. One thing that's nice to do is to make you can make really easy plots in Excel to just visualize the data. Like, so if you've got data on like your yield, you can just plot that against the different varieties and you'll kind of see what the yield looks like and how it compares. And very easy functions to do are just to do an average, like an average of yield across these 3 replicates. And if you're doing averages, you also would want to probably do standard deviation, which is again a very easy function to do in Excel, just equals STDEV, and then you select the 3, the values of the yield. And that just gives you an idea of how much variation there is in your plot. So if you've got an average yield of, 4, but that's because some plants have a yield of 1, others have a yield of 9. Like, you may not be very happy with that variety anyways. And so the standard deviation just gives you an idea of how much variation there is and how uniform your variety is. So here's an example of just some data from loosely Chinese cabbage trials at Everdale. And so here they took weight as a measure of yield. And we're looking at 5 different varieties here. And so you kind of see, like, it's a really very easy thing to plot. And these little black bars on top are error bars. So those are showing kind of how much variation there is among plants to get this overall yield. And so, I mean, just looking at this, you see some have slightly higher yield than others, but there's nothing that necessarily stands out dramatically. So, you could think, well, based on yield, like, you could probably grow any one of these. And then the question becomes, well, which one do you want to grow, like, for other reasons? And in the case of this trial, I think the most useful information came more from just, this fairly simple, more like observation. Like, you've got here, the 5 different varieties where the seeds came from. And then just an overall impression, like 9 being the best, 1 being the worst, like, and then kind of a verbal articulation of what this impression is. And based on that, I feel like this trial comes out with kind of a clear, kind of a clear winner of a variety you'd want to at least grow again, because, it says like good size, beautiful color, very marketable, good taste, would grow for sale again. And you know that there wasn't that much difference in yields among varieties. And, whereas if you look at the next variety down, it says like, by the time they fill out and are heavy enough for harvest, they're too large for customer preferences. So maybe you just let that one go. And then you've got these two others that seem, to have found some favor as well. Good taste, slightly smaller. So, maybe those would be worth trying again just to see if you like them the next year or not. And so that's kind of how, like in this case, the most useful data is actually very simple to collect, but it did rely on, going out and consistently observing the varieties. And once you finish your trial, you're going to want to make decisions and maybe set up some new trials. And so you might decide just to repeat the trial because plants perform differently in different years depending on the season, depending on the soil. And so you might not be sure. Like if you're looking at really like switching a variety you currently use for a new one, you might want to repeat it again before you make that decision. You'll probably want to retry certain varieties, discard others that just had no interest at all. You might decide, and I'd say especially if you're doing work with gene bank varieties, you might want to keep the very best individuals that are interesting but maybe need a bit of work for breeding a new population. Just let them all cross and select out of that and start your own breeding project. Or you might just change the emphasis of your trial based on observation. So like in my case of my little backyard spinach trial, now I've become interested in overwintering spinach. So, maybe you'll notice something that you didn't expect that will lead you to a new trial. And I'd say that would kind of conclude the cycle of doing a trial in 1 year. And, wish you as you, go about trying to set up trials, happy trialing.
Excellent. Thank you so much, Helen. Thank you so much for sharing all that info and some of the results from last year's trials. What we'll do before we launch into Q&A is give Rupert a bit of time on the floor in case he has any other tidbits or perspectives to share from the perspective of a farmer and a seed grower out in the field doing variety trials.
Hi, yeah. One major thing, Helen did touch upon it, is that a lot of trialing and information on different varieties is from conventional agriculture. There's not a huge amount of wealth of information out there on open pollinated organic varieties, and the trialing is becoming really pertinent to developing varieties that stand up well in organic or agroecology farming systems. So this is I think one reason why we're hearing more and more about trialing. It's becoming really, really important. And I would encourage all market gardeners, farmers, seed growers to start really entertaining the idea of trialing. I know one seed grower on one of the islands here in BC who was always dabbling in growing broccolis as one of his things. And he was always, occasionally trying this variety and that variety until one day he found a variety that just blew him away. And that's the only thing he'll grow broccoli-wise now. So the trialing thing, even though it can be initially a little bit of a chore to bring the whole kind of essence of trialing into your daily life. I know it can be a bit of a pain sometimes to be doing the record keeping, observation, you've got so much else to do, but in the long term it actually, from experience, is really beneficial. And also from a point of view of the fact, even though we still have people on the planet who don't believe that the climate is changing, it would appear that our climate is changing. So trials are becoming really important from that point of view as well. And with the climate changing, we're seeing slight variables in length of growing season, number of frost-free days. We're also seeing changes in dominance of different pests and different diseases. Diseases that haven't been an issue before are becoming an issue. Pests that we haven't had, that weren't pests before, are becoming an issue as well. So trialing from that point of view is becoming really more and more important. And I mean, there are other things as well, from a grower's perspective why trialing is a great thing to be doing, and to, as I said before, really bring it into your life as a farmer and a grower. It's going to really benefit you. Finding a new variety, as I mentioned with the grower who found this awesome broccoli, finding new varieties, can increase your economic security. It can increase your standard of living by finding a variety no one else is growing that you've trialed out and you found in your area. It just grows amazingly well. Another thing as well with trials and record keeping is keeping information in the public domain. It's becoming really important. The more information, the more record keeping, the more trialing of open pollinated varieties there is, and the more those records are shared within farming and growing communities, and that information is up for public review in this age of patenting or patenting. To have that information in the public domain is really important. The more information there is out there about different varieties, the harder it becomes to patent certain traits and certain varieties. That's a really important point, and becoming more and more important each day. Some things that you can learn, learning from trialing, again, that's going to increase economic security is looking at different varieties for sequential sowings. Some lettuces are prone to bolt when the heat starts to hit in the summer. Other varieties just love the heat, and so you can, with experimentation and trialing of, say, lettuces, you can be growing lettuces throughout the season, as opposed to growing them spring, allowing them to go to seed, and then planting again for market garden later on in the summer. You can actually be growing all through the season with different varieties of lettuce. So that's kind of mainly it. But again, just reiterating that this variety trialing is also a way of networking with people, to be comparing notes with farmers and growers, and where you're sourcing your seed from. If you're doing trials, as Helen mentioned, with various different sources of seed, you're going to start to pinpoint sources that are extremely reliable seed sources, which is something that's so important for market gardening farming. If you're making your living from things that grow from seeds, it's so important to be getting good, healthy seed. And with the trialing, we can start to be identifying these sources. And also, just as I said before, just this exchange of information amongst farmers and growers. It's so important. A little snippet of a variety that, like this gentleman who was the broccoli, I'll go back to that. The fact that he shared this information about the broccoli he's growing with so many people now has helped some farmers and some growers, market gardeners, to be producing a good crop of healthy broccoli, which again is, increasing their revenue. So those are just some of the points that come up with trialing. And again, as Helen said, happy trialing.
Thank you. Thanks, Rupert. That's incredibly valuable advice, especially in the context of the importance of trials in organic and agroecological conditions, and the importance of understanding just how strong of a networking and knowledge-sharing tool trials can be. We've got a few questions from the audience, and we can do our best to address them. One of them might have already been covered, but both Jen and Rebecca were asking about variety trial record-keeping templates, And Helen, I'm assuming those were the ones from the OSA that were on the slides, and there's been a request to put that link in the chat so folks can copy the URL. So if you don't mind doing that, one of us can do that, but do you have any other templates that you'd recommend for folks that are interested?
Yes, yeah, I'll get the OSA link, and like I think the OSA ones are very general, like you could adapt it to any crop. I'd also be happy to post the templates I developed for Chinese cabbage, like sort of head type Chinese cabbage, loose leaf Chinese cabbage, spinach, and I have one as well for like winter spinach for BC, like kind of grown in a tunnel, which hasn't really been taken for a test run yet. So I'd be happy to post those, I guess, in the same place as we post the webinar video. I think that would make sense. And just with the caveat, like that they were fairly specific and maybe too detailed, and you might want to definitely want to go through them and really decide kind of what data you want and which columns you would just eliminate.
Yeah, that'd be super helpful.
But yeah, happy to share it.
Great.
So I can send those to you and Steph, I guess, and they can go up.
There's a great document report from Organic Seed Alliance. It's called OnFarm Variety Trials, a guide for organic vegetable, herb, and flower producers. And I believe it has a whole bunch of templates and forms on it.
Yeah, it'd be good to put those up to accompany the webinar video when we post these. Great. There's another question here. It's coming from Genevieve. How many times would you recommend repeating trials?
I mean, it would depend. If you mean like years, I mean, I would suggest like probably 2 to 3 years. Like if you're really looking at like something that you're going to be growing, like that your livelihood is depending on to some extent, I wouldn't base like, a massive switch to a new variety based on 1 year of trialing. Like I think if it's good after a year of trialing, maybe you grow like a bit more of it the next year, but you might want to do a few years. Like normally like in kind of formal research, like it's 3 years of trials. I'd say at least 2. And if you're talking about how many times to repeat it, like how many times to repeat a variety within one trial, again, like my rule of thumb is I like 3 as a minimum, but like you can get away with 2. And again, it depends like how rigorous you really need to be. Like if you're just trying to decide if fundamentally you like something or not, maybe you don't need to repeat it as much. If you're really trying to get at like which of these varieties has the best yield, then you'd kind of want to repeat it, a few times and really measure the yield well and make sure that you're getting the right information, because that's going to have a direct input on how much you can produce and how much you can sell.
Great. There's another one from David Baldwin. He's asking about accessing germplasm. How difficult is it to get plant varieties from Canadian public gene banks? Is it only available to professional and university government researchers, or is it more accessible to farmers? And I guess it's a question regarding the access of germplasm from ex situ conservation.
I mean, my understanding is that, so I've always approached them as a researcher in some capacity, either through McGill or through USC. I would say USC Canada doesn't have the same research stature as McGill, for example, and I saw no difference in how I was treated between those two. And I think that, and I do know farmers who've accessed germplasm from PGRC as well, from the Canadian Gene Bank, or from the USDA, from GRIN. And really, like, their criterion and their job is to conserve this germplasm and make it available publicly to people who want to do research on it. And that includes farmer research. Like, that includes a farmer who wants to try it out for his or her farm or conditions or whatever. So, like, the important thing is just in your application, they'll usually ask you, like, why you want to use it. And, like, the reason you give needs to be a research type reason, not necessarily a very complicated one, but just to see, I'm looking for, a nice spinach, a nice fall spinach or something. And rather than a reason of like, you know, I need some seed to grow to sell at market. Like it, you know, there has to be kind of that step of you're going to be trying to document it, but there's, at the same time, there's no expectation that you're going to send any data back to them. Like they're kind of taking it on faith that, you know, you're contacting them out of interest for research and you're going to do the research on your farm, but there's no obligation. You don't sign any documents. You know, if your research doesn't work out, it doesn't matter. So, I mean, at least for those two, like USDA and Canada, especially Canada, like they're very quick to respond. They're very friendly. I don't think there's any problem with farmers getting germplasm. I've never heard of any.
Have you had much interaction with the Plant Gene Resource Center in Saskatoon, Helen?
Yes, yeah, they're the ones that I was referring to as far as the Canadian Gene Bank. And like I said, they're really open. They really want farmers to be using their materials. Like they have a strong relationship with Jim Tournier in the prairies, who like actively grows out a lot of their cereal materials. And they're pretty quick too. Like the USDA Gene Bank like takes maybe 4 or 5 months to get stuff to you. So you want to contact them like in, you know, October or something if you want stuff for the spring. Whereas the Canadian materials maybe take a few weeks to get to you, is my experience. So they're much quicker turnaround.
We've got time, I think, for 2 more questions, which have already been posted. One is on some thoughts or recommendations on how to build a routine on fitting regular documentation into the busy life of a farm, and what I assume is a follow-up to it is how often to do that documentation. I guess both of you can speak to this.
Yeah, I mean, I think maybe Rupert, maybe more than me, but I think the key thing is to make sure it is built in, like not to just to leave it as, like once everything else is done, I'll go document the variety trial. Like this is like an issue that happens in on-farm research, and it's very understandable because you have to prioritize the crops that you're growing, you know, for money as well. But I mean, I would say like kind of once a week can be good, just in terms of setting up a regular routine. Like if it's every, you know, Wednesday afternoon that you have a bit of downtime, like you plan for it then. Or every 2 weeks, but kind of like I think if you have a regular day where you're doing it, that helps. But I mean, maybe Rupert, as like a more, you know, more of a practicing farmer than myself, would have some tips as well.
Discipline. It is a difficult thing to get into the habit of, for sure. But I mean, even just recognizing or acknowledging the fact of the importance of doing it, and that in itself can be hopefully enough to spur us on to do it. One of the things is to have a dedicated notebook that fits in your pocket, which sounds kind of basic, but if you have it in your pocket at all times, then you know you can, when it strikes you need to be doing it, you can record in that. The beauty is as well, we have electronic gadgets that work in the field. You don't have to have a plug-in to use them. So, you know, having an electronic device on you where you can record data, which probably will have a camera on it as well, which is even better. It is a hard thing to get into the habit of doing, and I think this is why that we've been trying to really encourage people to do it, and it's like anything in life. Once you get into the stride of it, incorporating it into your life, then it just becomes habit. But it's actually adopting that discipline that can be difficult. So as I said, a notebook is a good thing, or an electronic device that you have on you at all times. Then there's no excuse because you have them on you. So, yeah, I mean, that's yeah.
I would say also, like, a lot of people who do trials, like, if they have some interns, on the farm, like they'll kind of delegate managing the trial to one of the interns, like as a learning experience as well. Like that can be one option. And I'd say also like having the trial close to where you frequently pass by, like as opposed to at the very far end of your furthest field, so that you have more occasions to be walking past it and so that you remember that you need to be checking on it is very helpful. And for the e-device, I'd say, like, I agree, like, I personally, like, I'm really, I don't know, I just like paper, and I find I always get my devices, like, all muddy or whatever. But if it's something that works for you, the huge advantage is that you then don't need to enter your data into a computer later. Like, it's already done. So I see a lot of advantages to doing that.
And just adding to that, it's like if somebody is given they're given that task as part of their kind of daily routine, it's like that's their thing to do. It becomes part of their job description. Then I mean that's an ideal way of bringing it into the structure of running the farm or garden. So yeah, making it somebody a part of their job.
Yeah, like not expecting it to just, like in budgeting, you know, it takes like 2 hours a week or whatever it is, like not trying to make that time come out of nowhere. I agree. It's part of someone's job.
Excellent. Those are some great tips. Discipline and integrating it into a core part of the farm routine, and not seeing it as like an external thing to the farm, but as an integral part to improving the farm operation is probably the best way to sort of prioritize it and figure out how it can be integrated into your system. We are unfortunately out of time for today. There are a couple questions that were left unanswered, but we'll do our best to get back to those participants and share that information with you guys. I'd like to thank Helen and Rupert again for taking all the time to share what they know with us. And we will absolutely keep you folks posted in terms of when our next webinars will be ready. As we mentioned, our webinar schedule will be available at seedsecurity.ca on our webinar tab. And the next one is Managing Disease for Solutions for Your Seed Crops on July 22nd, and you'll be sure to find out information about that soon. So thanks again, happy trialing, and good luck to all of you this farming season.